In this video, we will discuss about the experiment that has identified some new pigmented genes. So, we will continue our discussion where we end in our previous video. That is, they have identified some of the genes that might regulate the pigmentation in the human beings. And what they did is they knock out of those identified genes, they knock out some of their best candidates using three different guide RNA against each gene, and they can see the effect on the pigmentation compared to the wild type. That is, knockout cells are completely tyrosine knockout cells are completely lack of pigmentation compared to the wild type, and the other cells also have different degree of pigmentation. However, in each one of them are lower than that of wild type. Then they quantified the melanin content in different knockouts, and they find out that in all the knockouts the amount of melanin is low compared to the wild type. However, the degree of melanin content in knockouts is different. This might indicate that different knockouts will affect differently on the pigmentation. Then thereafter, what they did is they did the differential analysis. They analyzed the cells. They analyze four knockout cells system, that is tyrosine knockout, COM-MD3 knockout, KFL6 knockout, and WDR81 knockout, and at different stages of differentiation. And simultaneously, they also took the uh, electron microscopy images also, and here the electron microscopic images are also denoted at different stages of the cells. And when they quantified, they are able to see that in case of tyrosine, the stage 3 and stage 4 melansome stage 3 and the melansome stage 3 and 4 is very less. So the tyrosine knockout might affect the melansome at the stage 3 and stage 4, whereas for the stage 2 there is no decrease has been observed. That is, it might point out that at a later stage of melansome melanosome differentiation, the tyrosine knockout cells have less pigmentation, or their pigmentation differentiation begins in the melanosome stage 3 and 4. Or another way we can say that tyrosine works during melanosome stage 3 and 4. And for COMMD3 we have observed this thing only for the stage 4, and for KLF6 we can see the increase in at stage 3. However, the final melanosome stage 4 is decreased. And for WDR81 knockout, we see the similar results, that is, at stage 4 there is a decrease in the melanosome uh, differentiation, uh, and these things are also in line with their electron microscope images. Thereafter, what they did is they pick up two candidates, one is KFL6 and tyrosines, and identify and do a deep analysis on KFL6 knockout, uh, cons considering this tyrosine inactivation through this CRACE recombinase. So this is the image for KFL6 knockout only. In this case, we do not see any change in the pigmentation. One of the reason could be that tyrosine is compensating the KFL6 knockout. So we inactivate this tyrosine with the help of CRE recombinase that targets this tyrosine gene. And after that, we tested. After that, the researchers tested it for KFL6 homozygous knockout and KFL6 heterozygous knockout. And for the homozygous knockout, we can see the decrease in the pigmentation, and even the loss of hair. We have seen decrease in the pigmentation in the hairs. We have seen, and however, for this heterozygous knockout, we see very less decrease in the pigmentation of the tails, and also some spots only has been observed in case of tyrosine. In case of KFL6 heterozygous knockout, thereafter they decided to check whether this is because of the decrease in the melanocytes, or it is because of different it is because of their effect during the differentiation. And so when they did this analysis on when did this immunofluorescence assay against this melanome A, they can see that melanome A in the double knockout, and in the case of this homozygous knockout, and also in the case of this homozygous knockout with tyrosine inactivation is almost similar, or just there is a slight decrease in the homozygous knockout. So from this, whereas there is an increase in the double knockout and tyrosine inactivated uh, system. So from this, they made a conclusion that the KFL6 does not affect the melanocytes degradation. However, it will affect the it will affect a differentiation of these melanocytes at differentiation of these melanocytes at different stage. In that case, in KFL6 case, it is in stage 4. However, there is no effect of melanocytes, as melanocytes are almost comparable, or we can say that there are slight increase in the KFL6 dual knockout, and in which tyrosine is also inactivated by Crace recombinase. And this is the negative control where there is no melanocytes. And same thing they have observed using electron microscope images, that in case of tyrosines inactivated KFL6, the melanocytes are comparable to this KFL6 are comparable to this KFL6 homozygous knockout. The increase in the dark spot could be because of increase in the dark spot are because of tyrosine is compensating the KFL6. However, the melanocytes in both the cases are looks almost similar. And, the melanocytes are in this case denoted melanocytes are in this case are denoted by this small uh, vocule type thing, and this small vocule type thing, when they quantified this same image, are almost similar. There is no significant difference. So thereafter they decided to check whether there is this uh, knockout of KFL6 will affect or lead to any differential expression of genes or not. So for this, what they did is prepared they use these MNT1 cell lines. In this, they knocked out these KFL6 genes and and grow a single cell colonies of this KFL6 knockout. So, and they use three different kind of cell system. First, uh, in all the cell system, first they tag this KFL6 using this tagline FKBBV. So what the function of this tagline is that when someone adds the degradation tag, it will kill off this KFS6. So they use three kind of system. In one system, tagged both the they made a homozygous KFL6 tagged cell lines, and in another system they made a heterozygous KFL6 FKBPV tagline. In the last case is the similar to the first case. However, the treatment is different. In the first case, they treated it with DMSO, so it will not affect this KFL6, as it is not a degrading protein that will degrading tag that will degrade this proteins that is involved to FKBP tag. So in this case, there is no knockout of KFL6 happened. So this is act as a control. And in the second case, in the heterozygous KFL6 tagged, when they knocked out when they use the degrading tag, they can see the 50% decrease in the KFL6. And lastly, in the case of this homozygous KFL6 tag line, in the case of knockout this degrading tag, in the case of this degrading tag, it will knock out all the KFL6. And thereafter they did the combined analysis of differential gene expression, and they find out that 900 genes are differentially expressed. They did the RNA-seq of all these samples and then compare the control with this knockout, the 50% knockout and the whole knockout, and present the combined results. In the combined results, they can find out that around 900 genes are differentially regulated. Out of this, 169 genes are related to the melanocytes. However, there some of them are mentioned here. It is tyrosines, tyrosine, SAFB, CHD8, and some of the genes are also upregulated, such as VPS29 and PSEN. So this will indicate this differential regulation will indicate that the knockout of this KFL6 not only related to the this KFL6 not only related to the pigmentation of the not only related to the pigmentation of the uh, system, but however it also has other functions, other important functions that will get disrupted in case of this knockout of this KFL6 transcription factor. So I hope you have understood how this studies has been carried out and how they have identified some new pigmentation genes. If you need to understand this study in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.